So the title of my presentation is hormones, from hormones to harnessing the biotech revolution. How do we make the most of projectile biopsies? So why do we care about projectile biopsies? Well, it turns out that these samples are the most important way to acquire biological material from cetaceans. It's easily the way we acquire the most samples, biological samples from free-range cetaceans. And so we've been doing this for about 20 years now. And in that time, places like the Southwest Fisheries Science Center have also amassed quite large collections. So we have on the order of tens of thousands of biopsies covering maybe 50% of, or just getting close to 50% of cetacean species. So large collections. These are easily targeted samples that we can acquire relatively quickly on the scale of dozens, and in some cases up to 100 samples in a particular day. So this is in a field that's dominated where the rate-limiting step is acquisition of samples, especially when you're studying the biology of these animals. This is one of the most efficient ways to get biological material. Though I am really excited about the new stuff they're doing with capturing belows. That's a very neat thing. So even though we've collected tens of thousands of biopsies, it's still very difficult to get each one. It is, like I said, the rate-limiting step for almost any biological study that we do with cetaceans, free-ranging cetaceans. And so they're very valuable samples, and of course they impact the animals. So the more that we can do with a single sample, the more we make of these difficult-to-obtain samples, and we're minimizing the impact on the animals. And this is a figure I put together when at the turn of the millennium, when I just started grad school. And this was all the data that I kind of envisioned that we would get, and we're getting at this time from a single, that we could conceivably obtain from a single biopsy, or that was being obtained from biopsy. And each one of those kind of outer circles is the genre of markers, and then the information is on the final spokes there. And the intention here is to show we're still getting kind of the same type of data, but what we've moved forward in is we used to get predominantly just DNA from a single sample. But we're doing a better job, and I think this will continue to get better, is that we're now getting DNA, contaminants, hormones out of a single biopsy. And I do think within the next decade or two, we will be able to, as we micronize the procedures, need less material, we'll be able to get all this information from a single biopsy and then give a lot of basically biology for a single individual. And I think that'll help our studies quite a bit. My little focus has been this corner right down here, looking at the hormones, and that's what I'm going to tell you a little bit about first. Just give you an example of what I'm doing, and then I'm going to tell you where I think the field is going to go. So we acquire most of all the hormone signals from the blubber that's attached to these biopsies. And so far we've looked at pretty much four types of hormones: progesterone, testosterone, cortisol, and thyroid hormones. And they're in different stages of development. Progesterone and testosterone fully developed, fully validated. Progesterone being the absolute most important of the markers that we assay for, because it tells us about female reproduction. And this is important as we struggle. Yesterday we were talking about human threats, as we struggle to identify the threats and how they might affect biology, or the population biology of the animals. This is one way that we can start to look at a marker of population health, and that's the population's ability to reproduce. So we can obtain indices of reproductive success for a population using the pregnancy rate. We also look at testosterone to tell us something about the age distribution within a group by looking at sexual maturation, and we can determine breeding season. Cortisol is a method that we have totally worked out as far as quantifying the levels, but we still haven't validated it. Unlike progesterone and testosterone, where you know for a validation animal, an animal that you know is pregnant or not, or sexually mature or not, it's very difficult to get animals that you know are stressed or not, or any gradation of stress. So it has been very difficult to to finally validate the cortisol assay. But it's important. The Navy is particularly interested in doing this, where they're interested in how sonar activity might affect cortisol levels, might cause a stress response. And this is the way, actually, with Dorian's help, we're going to try to be able to validate some of the cortisol levels. And finally, just recently, we started a project to start quantifying thyroid hormones. And these are indicative of nutritional stress, also lipophilic, so likely to be found in the blubber. And we are working with a number of groups, and including hopefully SWIRP, in order to look to see how different prey conditions, but also contaminant loads might affect thyroid concentrations. So now I'm going to take you through kind of the most important one, again, and that's the progesterone. And this is these are progesterone values from the blubber of common dolphins, or bee common dolphins. And what we found is that pregnant common dolphins have a whole lot of progesterone, on the order of 60 to 100 times more progesterone than their non-pregnant counterparts. And more importantly, we see, at least empirically, almost no overlap between pregnant and non-pregnant. In this case, there is no overlap. There's a fourfold difference between the highest levels we see in non-pregnant animals and the lowest levels we see in pregnant ones. That leads to a very easy marker that clearly distinguishes pregnancy groups. And then I also want to say that Marissa Petrego has a poster out, and she shows that the signal is very similar across a whole range of different species. And so that gives us hope that even species we won't be able to validate on will still be show similar types of traits where we'll be able to distinguish pregnancy. And it is really very simple. I like this. This is data that just came from Marissa to me just a week ago, and this was 25 animals following those dolphins that were part of a live capture event in Georgia. The group there wanted me to quantify progesterone levels. They knew the pregnancy state from basically from ultrasound, and it was really straightforward. I mean, without even looking, only having the information that we have right here, it was very easy to see who was pregnant. I just wanted to kind of highlight how a no-brainer this is, because there's such large differences between those that are pregnant and those that are not. Okay, so how do we use this? And I thought about this a lot yesterday. In this case, what we're looking at is how particular stressors, how they might affect reproduction. Okay? We have the stressor here is fishing effort, and we're looking at how that might affect spotted dolphin pregnancy rates. So just a quick background on the tuna dolphin issue. From the 1950s to 1990s, first staining for yellowfin tuna led to exceedingly high dolphin mortality. Northeastern spotted dolphins were taken down to about 10% of their pre-exploitation level, so that's 90% that they totally lost. The fishing industry changed their seining methods in the early 1990s to release the dolphins before they brought up the tuna, and of course observed mortality drops precipitously, I mean magnitudes. But for some reason, dolphin populations haven't shown any robust signs of rebounding. One thought is that if you take a particular dolphin, on average, an individual dolphin is chased and circled 8 times per year. There are places where that number can be as much as 50 times per year. You can imagine how this continuous perturbation might affect and inhibit reproduction. It may cause stress response, or just may break up mating enough that you get a depressed reproduction. So we started to look at that. We had 208 biopsies collected from the region, measured progesterone levels, and found that 11.5% of the group was pregnant. More importantly, we found areas where we found really high pregnancy levels and those areas with very low pregnancy levels. And so you can see actually in this northern area, we have it's quite high. 25%, this is all females, 25% of all females are pregnant. Very low down here, this is just at 3.4%, and then 10% in the very southern region. So we started looking at fishing efforts. This is the distribution of fishing effort. This is based on an algorithm that a colleague of mine wrote. And what it does is basically it adds up the number of dolphin sets that took place in the time right before a biopsy was collected. So we're looking at 70 days before a biopsy was collected, 140, 180 days before a sample was collected, and they can add up the number of dolphin beds that took place in the area, and they're weighted by basically how close in time and space that they occurred. And when we asked the question, of pregnant animals, are they exposed to the fishery different? What we found is, indeed they were, and it was quite dramatic, the difference. Here are the three windows I was talking about, 70, 140, and 180 day windows. And in each of the cases, the pregnant animals had much lower fishery exposure indices than the non-pregnant ones. So this is kind of, it's a little easier way of asking, a statistically easier way of asking the same question, are in areas of high fishing, do we see a lower pregnancy rate? So this was basically the same result you would expect if there was having a detrimental effect, and it's quite dramatic. So again, as far as how this relates to this region, the idea is there's a stressor, we can quantify it, in this case fishing effort, and then we can see a potential effect to the population, and this is reproduction. So the same could be done with sonar exposure, contaminant loading, harmful algal blooms. Any of those are potential stressors that we have the ability to measure, and then we can measure a population effect at the same time. So this is a good way, at least, to start at seeing how some of these stressors might affect population. So let me just, this is a radical change from what I was just talking about, because this, I think, in a few years, well, at least a few decades, will make what I'm doing fairly obsolete. So you can see what I was, marker by marker, one at a time, I would develop, validate, and such. With the advent of the sequencing technology that Phil was talking about in his presentation, it really opens up quite a bit of new possibilities, new markers, and instead of looking at one at a time, even tens at a time, we have the ability to look at thousands, tens of thousands, even 100,000 different markers in a particular run for a particular animal for a particular piece of skin. And so this not only gives you a lot of different potential data points to look at, but you can get signatures. So if it is something complex of stress. And in this way, especially if we're looking at the transcriptomics, or the RNA, the protein, and the metabolites, you get a full picture of what's going on in the physiology of the cell. And so this just kind of tells you what happened at different time frames. So RNA just right before the sampling, proteins a little bit before that, and of course the metabolites a little bit before that. So you get a window of what's going on in the cells at a particular time. And there are likely signatures on a particular event. And so again, we'd be looking at reproduction, we could look at exposure of different events, but it is heavily dependent on validating the samples. You have to have animals of known condition in different, so pregnant or not, exposed to domoic acid or not, in order to be able to use this approach. And I did want to say that, well, though well behind the medical health field, omics is coming to mammal research, and it's coming on kind of recently, but quite a bit has taken place in the last 4 years. The first dolphin microarray was published in 2007. It had on the order of 1,000 genes on it. In 2008, we got our first genome, bottlenose genome sequenced. It was a rough sequencing, but it was fully annotated. It was 2x coverage. 2009, we had our first skin cDNA library, and again, this is really important. Unlike the leukocyte database or microarray that happened in 2007, again, skin is our most likely sample that we're going to obtain. So it was important that they got a skin cDNA library, and soon they'll be constructing, or probably have already constructed, a microarray from that cDNA library. In 2010, a group of undergraduates at San Diego State University started sequencing and had finished sequencing the sea lion genome. It's being annotated presently, and we'll have that. And then there was an updated array another magnitude more of genes, and that will continue to take place. We'll get more and more genes in these microarrays. And then finally last year, which I think is actually most excited about, is the establishment of the Marine Mammal Omics, I said working group, but it's really a consortium that was put together, and it was a group of people with very different skill sets and interests, and it takes that much in order to get omics projects going. But all together, different resources, different capital equipment, together kind of pushing forward to the same goal, and I think that this will keep us on the same track. And I think what I'll do, I do have, this is the last slide here, showing some of the obstacles, sample quality, validation, large capital investments, and sharing of information. And what I'll do is that during the discussion period, maybe we can talk about some of these future obstacles. Okay, thank you very much.